Hi, good evening. It is July 31st, 2021, and I am actually just practicing giving this talk as a technical presentation coming up. And starting now, I'm just going to pretend that I am giving this talk and it is that day. But I thought it would be a good idea to record this just in case anybody wanted to watch it, or actually, if at this specific presentation anyone is missing and they would like to see what they missed. And so, with that, I'll start. Ahem. Hi, good afternoon. I'm Aaron. Today I'm going to be talking to you about microphotography on a budget, which is a topic that is dear to my heart. Just a little bit about my background. I have a bachelor's degree in biochemistry, and I did research on an organelle called lipid droplets, which is involved in storing fat in your cells. I also was a fellow at the Hebrew University in Jerusalem, where I studied nematodes. I have a master's degree in biomedical engineering at Lawrence Tech, where I'm currently an adjunct, where I studied microphotography, scanning microphotography while I was a master's student. And then as an adjunct now, I mostly study accessible methods for 3D printing metal, glass, and ceramics. So these composite materials. I'll talk a little bit about that, not much. A fun fact about me is that my dad put my name and my birthday wrong on my birth certificate. And this little picture is just having some fun. That's actually my 3D printed head back there. I made one for the whole department. It's a gigantic head that you can wear over your head as a mask. And this is one just of my friend Daniel. I thought it was kind of fun. So I mentioned that I have a biochemistry degree and I've done research in lipid droplets. So this is just talking a little bit about that. Um, I talked about lipid droplets, and one of my model organisms was heart cells, so myocardiocytes. And these are interesting cells because your heart in your body, your cells don't normally divide. That's why a heart attack is so destructive. And then these cells were unique in that you can see they actually undergo cell division. So that's what's happening. And then another neat thing about them is that they do beat inside of a dish. So that's very mesmerizing to watch. And so this is an example of microscopy, bright field, and then confocal on the left, where you want to see as detailed as you can. You want to be able to see the cells, you want to be able to see their organelles. Whereas this other work I did is kind of the opposite end of the spectrum. We still call it microscopy, but what I did here was I had this wide field video of nematodes, these little worms, in a petri dish, and I wanted to be able to capture a large area all at once so that I could track their motion. Each of these tracks represents a single worm. I wanted to be able to track their motion while they were exploring whether a food source was legitimate or not. And so very briefly, what I did is I put a small amount of a chemical in here. It might be actually popcorn smell. It might be the smell of almonds. It might be the smell of paint thinner. There are interesting smells that nematodes find attractive. And then I would monitor how long it took them over here to realize that it wasn't a real food source. So I would take the kind of the average of their positions and plot them like this and see, okay, with one chemical, it takes them this amount of time to eventually leave the area. With another chemical or another concentration, it might take them a different amount of time. And so this was a very, very, very pure research, not an immediate human application, but it was interesting about how their neurons could communicate with each other. And also when I came back, I used this as a model for teaching experimental methods for a biology course for 2 years. So I used nematode chemotaxis. Students would actually bring in chemicals from home, and they would monitor how the worm reacted to them, whether they were attracted, whether they were repelled, or nothing. All right, so now we can start to get into the meat of this talk. And so with these experiences, with microscopy looking very close as well as looking, kind of wide, I wanted to be able to make a system that could resolve fine detail but also do so over a wide field of view. And it was important to me that this was inexpensive because I had classrooms in mind. Also, that it was small and portable, capable of fitting in an incubator. I'm about to sneeze. Maybe I'm not gonna, edit that out. If I do, it's gone. Forgive me. We're continuing. So I wanted to make a one that was capable of fitting in an incubator. This was for both C. elegans, so the nematodes, as well as for cells. We didn't really end up doing that, but that was my goal, and it informed some of my design process. And then I also wanted to be flexible in choice of optics and stage and motion control. And I put a little asterisk over here for fine detail because I'm aware that the system that I might be using is we're talking about a few like tens of nanometers of precision, whereas the systems that I end up building are on the order of a few microns. So just about 1,000 times worse. So this is supposed to be a technical talk, so I am actually bringing in some of the background behind the problem with creating a system that can see both very far and wide and close up. And the problem is simply if you have a higher numerical aperture lens, this just refers to the angle of the cone of light that the lens accepts. If you have a high numerical aperture, which can resolve small feature sizes, then you also get a small depth of field. And so this depth of field is just the minimum, is the width where we call this a tolerable, we say that it's in focus. In reality, there's a maximum point of focus, but there's a range at which we consider the amount of focus acceptable. And this acceptable range decreases quite fast as you have higher and higher numerical apertures as you zoom in more. Similarly, aberrations, spherical aberration, chromatic aberration, pincushion, barrel, they become more and more apparent as you look over wide fields in the same image. And we can see that in this diagram, when you bend light through the edge of a lens, as you get more towards the edge, the light has to bend more and more, and the light deviates from the ideal location more and more. So we can see that one way to remedy this is to use a smaller image field, a smaller image plane, because the smaller the plane, the light comes from more towards the center of the lens. It's the same concept behind a pinhole camera, or if you've ever lost your glasses, you can do this, and it just forces the light to come straight on, where it doesn't have to bend as much by your lens, and so images become less blurry. And then in layman's terms, just the more you zoom in, the less you can clearly see at once. Macrophotographers know this, microscopists know this, but you really just can't get the most of both worlds. So while I was doing this project, these things, optical disk drives, they actually became a major source of inspiration. And it's worth talking about because they both are a source of inspiration behind why I do the things I do. And then they also are a source of parts. So this is a Blu-ray pickup unit from a PlayStation 3. It's kind of an anomaly because all of the parts within it are meant to be modular. It's almost like a laboratory prototype that was pushed to production just because they ran out of time. But it has 3 diode lasers, these precision voice coil motors, a very high quality 0.85 numerical aperture lens, objective lens, that's carried by these floating voice coils. And I actually got a bunch of these from a scientist, a laser scientist, who buys them just for a single beam splitter cube on the inside. So these cubes, if you were to buy them from Thor Labs, they cost $180 each. And so he sent me, a few dozen of these that just had that one cube removed, so I was able to play with them. So it's worth talking about the operational principle behind the disk drives. And you'll see that most of this talk is not about disk drives in this way, about this specific operating principle, but what I do is still analogous to them, and it's what originally got me inspired to work on them. And maybe it's obvious for people in the industry. This is how pretty much everything is done, but when I learned about this, it was very cool, very much like an epiphany moment. So the basic principle behind all disk drives, whether they're CDs or Blu-ray players, is you have a monochromatic light source, this laser diode. It shines through a polarizing beam splitter as well as diffraction grating. The diffraction grating splits the light up into many subbeams, which will be used later. So a main beam and then many subbeams. The polarizing beam splitter, it linearly polarizes it in this direction. It passes through a cylindrical lens, which it causes a little bit of astigmatism. Some designs omit this. They rely on the natural astigmatism of some lenses of the system. It goes through a collimating lens to focus into a parallel beam. It hits a quarter wave plate, which circularly polarizes it. And then it goes through the main objective lens, and it says it's a biaxial device. This is the voice coil motor over here that moves up and down and provides very, very fine precision control. When it hits the disc, if it hits a pit, destructive interference severely reduces the signal that comes back. But if it hits a land, it travels back to the same optical path. It gets circularly polarized again, but this time it's circularly polarized in the other direction, so that when it hits the polarizing beam splitter, it is reflected instead of transmitted. It then hits a photodetector array, and the array uses the astigmatism caused by the cylindrical lens to tell whether the object was in focus or not. And it does this through this little scheme here, where depending on where the beam is, if it's perfectly focused, all of these 4 photodiodes will be illuminated. That's this one in the middle. This is an actual image of one of these arrays. And then if it is out of focus, A or C or D and B will be illuminated. And you can tell whether it's too close or too far, and then you can correct for whether it's in focus or not through this method. So I wanted to show you this to talk about 3 main points. One, they solved the problems before of small depth of field by using an active feedback method. So they tell whether they're in focus, and then they adjust their system to make sure that it is in focus. So they use some kind of active feedback mechanism. They solve the problem of aberration because they're using a very high numerical aperture lens. They solve the problem of aberration by using a very small depth of field, a very small field of view. They have a pinpoint laser. And they also use a pinhole size detector. This is a very, very small detector, and any out of focus light or any light that scatters does not reach the detector at all. This is actually similar to a confocal microscope. And additionally, finally, they solve the problem of small field of view that they just created by using a very small field of view. They solve this problem by physically scanning across the disc with motors. So the disc is spinning. And they're using this stepper motor. The disc spins here, and they have the stepper motor, and the optical pickup unit moves forward and back. So they physically scan across the disc. They replace, instead of trying to image the whole thing all at once, they settle for being able to image just a little piece of it, and then they physically scan across the disc. Okay. So my strategy at first, I wanted to try and make something with a spinning dish. So I actually did try that, and it's kind of a hard controls problem, especially if you don't have. So the disc has lines to keep you in place on the disc itself. I didn't have that on like a petri dish. So instead, I tried to make a linear scanner. And so the basic strategy behind this, it's pretty simple, is you just take, you use the whole disk drive, and this motor that carried the optical pickup unit, you either use it as it is, or you replace it with like a camera or a digital microscope. And then you have a perpendicular axis that has some kind of stage. So maybe it holds a cover slip, or it holds a petri dish or a slide. And then you can cover the whole area, and it doesn't matter how limited your field of view is, you can cover the whole area. So this is just a little bit, you can get a glimpse into my thought process, my design process. So I'm very much an experimental person. If I have an idea, I want to try and get it implemented as quickly as possible so I can see what kinds of problems I'm dealing with, and then I can later optimize them. So I actually used glue sticks in this case. I'm trying to hide the taskbar. All right. So I actually used glue sticks in this case, where I tried to just arrange the disk drives relative to each other just to see what they look like, and then I saw that okay, an inverted setup is not going to work out. So I made this upright setup. This is 3D printed, and you can see like I used curved legs to accommodate the structure of the frames, because this was all about reusing as much as possible, to keep it on budget, not have to make as many parts. Just a little bit more about the physical design process. So the digital microscope is the replacement for the pickup unit in most cases. So it snaps in here. It's an annular snap fit, very optimized for 3D printing. This prints in about 20 minutes, no problems on any printer. Even uses the original switch. And you can see that I actually, instead of putting the digital microscope directly in line here, I have it floating, and that lets you adjust it. And it also prevents any push on the motor, prevents any strain on the motor. And this is just plastic, but I do have some repeatability data comparing this plastic unit and the original unit on supplementary slides if you would like to see it. So there goes the two axes, the microscope one and then the stage. And one thing I would like to do is I'd like to add a z-axis. So in this implementation, I actually am using one of the motors from inside the Blu-ray player. So this z-axis motor is actually normally used, it's adjusting the collimating lens back in this diagram here. And in the Blu-ray player, it's adjusting this lens to be able to not focus on the disc, but to switch between layers when using when scanning a multi-layered disc. So this motor, it's very precise. It's for me. So 1 micron precisioning over a 3mm range. It has its own little optical interrupt switch in it for homing. And so this ended up being my z-axis. But you can see, wiring, by hand, it's not super easy. I did come up with a better solution though. That better solution is this little PCB board. So I do have some amateur, PCB design experience. And I also added, instead of just lifting up like a cover slip in this previous slide, we're lifting up an entire rotary motor. Okay, so we're lifting up an entire rotary motor, and now we can spin around an object as well as just lift it up and down. So now we have 4 axes of rotation of freedom. Here on the left is an example configuration. So this is, a complete system. So I meant, portable and small, fit inside an incubator, even though we're not really doing that in these demos. A Raspberry Pi controls the imaging and, the motion control. I use just easy drivers to move the steppers. And then actually in this implementation, the motion control is at a very low level. So I'm actually telling the motors exactly to move, how many steps and, controlling the timing and the homing procedures. Whereas in a later versions, I actually switched to a G-code based system, if that means anything to you, where I let kind of pre-written software by other people handle a lot of the back end. And then, for all of my systems, I usually have some kind of, graphical interface. I do most of the scans, programmatically. So, just because I know the system, so I'll tell it exactly what to do, in the Python program. But, here's an example of one of my little graphical interfaces. Here's another just quick showing of the complete system, showing motion control. And that's the camera one. And again, we're not really talking too much about it, but I have done a lot of experimentation with the Blu-ray systems. The results are more raw, so I'm not going to be showing too much of those results. But just to show you here, in this spot that I'm circling with my mouse, this is where the original photodiode detector array was. Rather than tap directly into that at the beginning, what I did instead was I replaced it with an external photodiode. And then I have just like, a regular transimpedance amplifier to boost that signal. And that's connected through the GPIO in my Pi. And then I'm scanning in this system. I'm using the reflectance to try and form an image of this little piece of paper underneath it. So I have just a test pattern, and I'm trying to determine. I'm trying to scan it basically. All right, so I'm going a little over time. Again, this is a practice run, so it's okay if it goes a little bit over time. I'm trying not to get past 40 minutes, but now we can get to the results. And so first, here's an example of some laser imaging results. Not very impressive, but there on the left is an original, and there on the right is a point scan. And you can see that it's, qualitatively, it's the same image, but it's pretty horrible. And there's many reasons why. I'm happy to answer any questions if you're interested in that. One reason is that external photodiode. It's probably not aligned. And also in the original Blu-ray or disk drive system, it's expecting part of the disk as part of the optical path. So many reasons for this. It is possible to get good images with this hardware, and eventually I would like to do that. So now we can talk about the camera-based systems. So I said that this is very analogous to the disk drive. Instead of using a point scan, we're using a very small field of view. These cheap USB microscopes have a tiny sensor, 0.3 megapixels. So that's actually a lot of pixels, but it's tiny to most people when they think of megapixels. And so we can see here that any individual picture does not cover most of the field of view. This is a relatively low magnification, but we can see that we can stitch together many of these pictures into a full image. And we can also do this repeatedly while rotating our image. So while rotating, we get just a bunch of fields of view. We scan 2D for each one, and then eventually we end up with this three-dimensional image. Here's just a couple more of those, insects. I was interested in insects at the very beginning because they have a lot of interesting geometry and color, and they're found, dead and for free in swimming pools. The idea of stitching your images, that can be done if you take images manually too. So on the left here, I actually used this digital microscope to just manually try to cover the whole field of view and then stitch them in. This was in commercial software that can align or register the images. But we can see that this automatic scanning result is clearly superior. I was kind of blown away. This was the first image that I took with this. You may ask about 3D model generation because I do have this three-dimensional field of view. In this instance, you can use it to create 3D models through the method of photogrammetry. And then photogrammetry is a blind method that uses, it looks for matches between images. It tries to find registration points between images, and then it calculates the hull, the shell that would be created if they were all properly matched up. But this method is not optimized for it. For one, photogrammetry assumes that the object is stationary and the camera moves around it, whereas in this case we have a moving object and a moving camera, and we're doing stitching. But it is possible to still, try to trick the model into giving you a 3D model. I made the discovery that, not really a discovery, but that polarized light is really great for revealing the detail, especially of shiny objects, but also for objects that are relatively flat but have subtle variations due to like fractures or surface stress. So this is just polarizing my light from the microscope so that only light that is itself polarized will that itself distorts polarization will reflect. So this is a ceramic casing from the chip of an optical mouse, and we can see that there's a real world in everything that we don't really realize. One of the problems, as I mentioned, was that the Blu-ray player solved was it used feedback to correct for focus. And so, that's what focus stacking is we are using information about which pixel is in focus. The method is that most use is the variance of the Laplacian. So it actually transforms the image into the frequency domain, and then it looks for the correlation between neighboring pixels in that domain. And the basic premise is if the pixels are highly correlated from one pixel to another, then that generally means that the image is blurry. Because when you have a sharp image, the pixels tend to vary more dramatically in neighboring pixels. And so Z-stacking is actually quite analogous to what the Blu-ray player does. It's taking many images, or it's looking at many heights, and then it is doing some kind of analysis to see whether each place is in focus. And then it's picking the part that is in focus, and that's what it's keeping, and that's the information that it's saving. So this is a ballpoint pen using external software. But the problem with, as we've shown, using external software is that we have a kind of an image pipeline, maybe stitching, stacking, 3D model generation, and it would be great if we could keep that all in the same place so that we can have the parts communicate. We can combine stacking and stitching even over large areas. I apologize for this low quality GIF. The actual images are quite a bit better, but we can see we have a whole RAM chip in this case, and we're able to eventually zoom in quite a lot of detail onto one of these. I hate to say whether it's a resistor or a capacitor, but we can zoom in into a very small point, even over what was originally a large area. I might change this slide in the actual presentation. Doesn't look so great. So I just mentioned the idea of Z-stacking. So here is our own homebrewed implementation of this is here on the left we have this Z sweep, so we have many images taken as we move our camera or our object up and down, and we can see that most of it is not in focus. And so to approximate the kind of standards of image stacking, all we do is we break the image into chunks, maybe 4 chunks, maybe 16, maybe many more. And then for each chunk, we mix and match which image is in focus. We pick the chunk from many different Z heights, and we pick the best chunks, and then we reassemble them into an image. So it starts out blurry, and then over time, it becomes more clear. And there's a couple problems with this, but it actually works pretty well, and that's kind of what I use now. And then the final image is pretty in focus. A great feature of this is that now that we have, when we do this image stacking, if we just keep track of which Z heights the good chunks came from, then we can interpolate the Z heights to some kind of grayscale value. So let's say your images are taken from 1 millimeter to 2 millimeter. You can say that 1 millimeter is black, and then 2 millimeters is white, and then everything in between is some grayscale value from 0 to 256, or 0 to 255. Python count starting from 0. So yeah, that's what we're doing is we take these little chunks. We keep track of the Z heights from each chunk, and we get a little area that's a little tiny depth map for each sub image. And then we combine many of these sub images together into a fully stitched object, just like our scans from before. But this time we're doing it not with the original image, but with the grayscale values representing the Z heights. And as you make the chunks smaller and smaller, we get something that appears to be our original image, but this is not just the original image. It's not just the original image in black and white. This is the original image according to the depth of every pixel. So this white area is closer to you, and then this black area is the furthest away from you. And there's some noise. So this is actually shot noise that I have an idea of how to clean up, but I didn't get around to by the time I gave this talk. Sorry. So this depth information is obtained for free. That is, you do this every time you create an image like this one, like this big image. Every time you do this, you also get the depth map with it for free. So this is a Roman Antoninianus, a couple thousand years old. And I mentioned I do metal 3D printing, and so I actually use this to kind of counterfeit this coin. So I scanned the coin, and my method of metal 3D printing, I'd be happy to talk about this. I don't really have time. But briefly, what you do is you print in metal mixed with plastic. So this is just like regular metal. This is just like regular 3D printer filament, but it's 90% metal by weight. So it just barely extrudes through the nozzle. And then you actually remove the binder, you remove the plastic, and then you put it in an oven where the metal actually sinters or shrinks together. And so through this method, I was able to make this copy of a Roman coin. I will now eventually bury it. I will bury it in an ancient Roman archaeological site and try to confuse the archaeologists. So this is just kind of like a little pet goal of mine. Is this on the left is the scan of the original coin. This is the original depth map. And then after printing it, debinding it, and sintering, this is what I eventually got. And so throughout that process, there was some kind of distortions or warping that's fairly typical. And I'm interested in the possibility of using this scan to do some kind of inverse compensation to see if I can like turn this into some kind of like kernel I can apply to the original image so that when I print it and center it, it'll be closer to this original image. And then finally, the last thing I want to talk about, because this is a applications engineering position, is one specific application. I had many, but the problem is that there are too many. Okay, I didn't even talk about all of them. I didn't talk about my automatic insect follower or my PCB scanner, but the scanner does too many things. And I found this out when I released my Kickstarter. So I actually tried to crowdfund money for this. I tried to raise money to turn this into a product last year. Not a good time, but I tried. I would eventually like to try again. And the problem was that the customers wanted the most advanced system. That was a nice surprise. But they also wanted it optimized for their specific use case. And I didn't have their specific use case. I had a billion use cases, and I said, I'll just make something that does everything. And then they said, well, I don't think I'm going to be able to know how to do that. It's going to be too complicated for me. It's too general. So, I talked with my customers. One guy was a photographer for Honey, I Shrunk the Kids. There were microscopists. More than one person who was interested in spider genitalia for some reason. I'm not incredibly sure why. But this was actually not the only person who wanted this specific thing. And then several people who were interested in coin photography. And that piqued my interest because coins are relatively consistent sized and shaped. They have a big customer base, and their customer base, the people who collect expensive coins, they also tend to have some money. And so I created this solution specifically for numismatists, trying to find an application. And then what makes this, specifically for them is this system completely automatically does everything for you. So it homes against the build plate, looks for out of focus objects, determines their boundaries, like so. It does a grid map to find where it's in focus and interpolates that. And then it starts the scan and it runs a complete scan. It runs a complete scan. It does all of the post-processing for you. And you get your image. You also get it in depth map form. So that's just talking about, my one example of I had this hardware and I wanted to find a specific application for it. And it looks like I am 38 minutes in, so I'm just in time. So, I guess I missed a slide, but this is just, I gave a presentation on this topic to the, Buenos Aires Numismatic Society. So I am no stranger to reaching out to people and giving these kinds of presentations. I'm very comfortable with it, even in other languages. And so with that, thank you very much. I'm happy to answer any questions. And yeah, thank you. And now I'm just going to go through some of my supplemental slides real quick. So this is, while in my optics engineering class, I wanted to simulate an optical disk drive. So this is just a, kind of a ray modeling of, going through this, this lens and then focusing again in the polycarbonate before it reaches the disk surface. Very cool. I can't say I properly understand everything in this. I was mostly following a tutorial. This was a ready-made system for me. This slide just shows that there are many, many implementations of the system. On the top left is a video showing the laser side of the research. So like that scanning, and some of the kind of hacks I took to reach it. On this right, here is a 3D printer version showing some of the motion control. So the 5 is a 5-axis version, and how I fix like offsets. So like when you tilt, you also create offsets in X, Y, and Z. So this just corrects some of those. At the bottom left, I have my auto bug follower video. So this is really kind of just for fun. But following an insect or following any kind of moving object in real time. And then I mentioned this on the bottom right. I have automated PCB inspection. So this is it's actually just going through on the PCB Centroid file for when you would put components on it with pick and place. It's just going through it, and it's just taking a picture of each one, finding the right path through traveling salesman problem. Here's another video, about a scan of an osteodensification drill bit. So just a specific use case. This is a biomedical one, because I did do biomedical engineering. So just showing you manual motion control. And then it'll show you my great camera ability, automated scanning, this rotary scan, Z scan, and then an X and Y. I mentioned, amateur PCB and schematic design. I wouldn't say these are great, but it was kind of a fun challenge to try and pack as much as possible into this 10 by 10 centimeter space that JLC PCB provides you. And also, it turns out that by sectioning this off, they noticed that I was trying to get a free board out of it, so I had to pay extra. Already showed that. Don't call the Secret Service on me. There's problems with this. But this is, you actually probably the only way you can scan a $50 bill, because if you put this in your regular scanner, there are these control patterns that will prevent you from scanning. And then you get a lot more detail than you can see here. So right over here, where you have this, maybe I'll make a slide of that. All right, and I've made a slide showing you this little part, and there it is. So you get a lot more detail than you have at the surface. Plenty to counterfeit, so please don't call the Secret Service on me. Oh, just another image about this butterfly. I had enough images though, I thought. Just a very jittery image, a very jittery GIF of following an ant around. This was stabilized. Not a very good job. It needs a control loop or two. I didn't show any code because most of the code is really bad. So there's a lot of spaghetti in it. But this is just showing you some of the path behind how blur is evaluated, and how like you get a bunch of you pick the best ones from a folder. So this one, you pass in a folder of images, like let's say all of one XY location but different Z heights, and then this will use the blur metric through the variance of Laplacian method, and it will pick the best image from each one through that. So it'll start with saying, okay, the best image has a blur of 0, and then or a focus of 0, and then as you get better and better images, it just replaces. It says, okay, this is the best image now, and then at the end of the loop, it'll save that best image. And then this is the same. So this is the same thing as stacking. You just do it to each chunk of the image instead of over the whole image. But you can I also do use this to like throw out very obviously blurry images, so I don't include them in the stacking process. So this is what happens if you mix and match. So this is at the very beginning. This, instead of before we did stacking. Oh, there should be scale bars. Ooh, this is, I'm going to replace that too. All right, there we are. So now we have, with the scale bars, just to show you. And again, these are very, 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 very much not zoomed in. So we have a couple micron resolution here, so we can zoom in a lot more into each of these little spots. But yeah, this is what happens when you mix and match images from different folders, depending whether they're in focus. And so we can see that, over areas that slope rather gradually, we can get all of it in focus now. But we're not going to get it in focus if there's like a point where there's a sudden change, like an overhang. So like right around here or right around here, you can't get it all in focus. You would need stacking for that. Oh, I did promise that we would talk about missed steps as determined by loss after homing. So this is comparing the plastic printed version that holds the camera versus the original Blu-ray assembly. And so I was actually surprised that if you just what we did is I told the stage to just like move in random patterns like a few tens of thousands of times, and then to home to go back and hit the switch, and then to calculate how many steps it was off from. So if it expected, that it moved, if it expected that it was at position 50, but it actually took 52 steps, that counted as 2 missing steps. And so it is true that if you do this method, maybe you move missteps in one direction and then in the other direction, so they cancel each other out. But in general, over time, it should walk to be away from the correct position. And so this is just comparing the original assembly versus the 3D printed version, and the 3D printed version, at very least, did not perform worse. And probably most of this failure is actually not due to missing steps. It's due to the switch not getting pushed exactly at the same amount of steps. So maybe the 3D printed version, it's a little bit more slippery, and it was able to push the switch more reliably slightly. But either way, it's not worse. This is not even relevant. This is just me trying to sort micrometeorites. So I'm trying to automatically identify micrometeorites from pebble and source material. So there's like a bunch of contamination, and I'm like, well, maybe I can look for sphericity. And this is a horrible method. Don't ever do this. You should try, if you're trying to sort them by sphericity, you should be trying to physically sort them by sphericity, not trying to just like image a bunch of junk. Try to physically sort it as much as possible, and then use a human. Another image of this is an Indian head coin. One of the pictures I took for the auto coin lecture. A variation of my scanner. This is just a version that uses where the camera is stationary, with the idea that you could then take your own camera and stack and stitch and rotate. And I still think this is a good idea. I still think that this would be a really great product if you could make it high quality and be able to sell it for like a couple $100. I think photographers would love to buy this. Software, though, that's another problem. Oh, more 3D printing and computer-aided design. I don't think I'll be doing any of that for this job. Yeah, and we already talked about this. And I think that's it. We've gone too far. So with that, that's everything in the talk, including the supplementary information. I don't think I'll get through the supplementary information, but it is there. So thank you if you did watch this and you are from this company. I'm very impressed that you sat through this. I hope you'll consider me for the position. And then otherwise, if you're just watching this because you're interested, and very happy that you are interested. And so with that, it's Saturday. I am going to go home and I'm going to eat some dinner. So thank you and bye-bye.